Okay, well, forgive me for zipping through these kind of quickly, but next slide. So we're going back to the moon and Mars. I don't know when, okay? Don't ask me when, but I know eventually we're going back there and we're going to build bases, and the bases are going to become more complex over time. Next slide. And our current way of kind of going on a picnic, bringing everything that we need to consume with us is okay in the short term, but I think there's a general consensus that as mission duration increases, there'll come a point where it's more economically viable to actually grow our own food. And plants have a number of advantages for bioregenerative life support systems. They scrub CO2 that we produce. They generate oxygen that we need. They can purify water through transpiration. And by the way, they also produce food. Next slide. So NASA has spent quite a bit of time with ground studies, working with hydroponics, and this has a number of advantages for plant management. And very high productivity. Some world records in terms of wheat and potato have been achieved. Next slide. At Kennedy Space Center for about 15 years, there was the agency's #1 test bed unit, the biomass production chamber that Bill Knott, John Sager, Ray Wheeler kind of oversaw, along with our contractors for years, growing all different kinds of food crops, potatoes, tomatoes, and such. In relatively closed environments, recirculating as much as possible. Next slide. NASA has also funded academicians, people like Bruce Bugbee at Utah State University, to develop specific strains of plants that are suitable for space applications, primarily small, highly productive, things like superdwarf wheat and also superdwarf rice. Next slide. And you can do, there is one way of doing a hydroponic system in microgravity. It's called the porous tube nutrient delivery system, where you actually have nutrient solution going through a tube, and plants will send their roots down. They're wrapped around the tube. I've made a slit here so you can actually see the root mass. They can extract all the water and nutrients they need. But for the most part, most investigators have grown plants in space in a substrate-based system, which will have a plant tray with porous tubes within to provide water, irrigating the substrate, arsenite or turfase or whatever, and usually adding a slow-release neutralizing pellet, something like Osmocote. Next slide. Some of the early studies with the SVET greenhouse experiment on the Mir space station, there were moisture sensors incorporated, and those data allowed people like Scott Jones and Danny Orr at Utah State University to come up with models of how the water distribution changes in the root zone of plants being grown in space. So in the 1G environment, you can imagine water is being introduced through these tubes, and you have a capillary mat that wicks some of it up to the surface. But gravity takes the excess water, just like here on Earth, and it pulls the excess water down to the bottom, so you're wetter on the bottom and you get progressively drier as you go up. Well, in microgravity, you don't have gravity pulling the water down, so you have an excessive amount of wetness in the middle of the root zone, and this actually has been a problem with a lot of the earlier studies. It's too much water where you don't want it, not enough water where you do want it. Next slide. Of course, the root zone doesn't just produce water and nutrients for the plants, but roots respire just like us, so they need oxygen just like us, and the zone in between the substrate particles has air with, of course, oxygen, and at 1G, you can imagine as your root zone gets more and more saturated, you fill up these pores until they're fully saturated. But in microgravity, that actually happens in an accelerated basis, where the water goes up around the pores, effectively sealing them off so that the oxygen that's within the pore space is no longer available. So often we've had a problem early on, especially growing plants with not enough oxygen in the root zone. Next slide. So one of the great values of having plants in space, eventually for food, but for the short term, it's this psychological benefit. Okay, you have astronauts, crew members who are up there in a relatively closed environment. There's a lot of smells, volatile organic compounds floating around, and they just love having a piece of Earth with them. This concept of the salad machine originated out at the Ames Research Center. The LADA system on the Russian side of the station is considered a vegetable production unit for generating some information, even some psychological information of this type. The Russians have another unit in development that they hope to fly in a few years called the Vitacycle or the phytoconveyor that will be a steady state producing salad machine, actually a real salad machine. Next slide. So I've just included this for some of the near-term and mid-term objectives for potentially using plants in space, and the amount of water that they process can reach 100% relatively quickly. Food is a little bit slower. Next slide. So when we talk about effects of microgravity on plants, you have to separate direct effects of microgravity versus indirect effects. So you'll see later on that the lack of convection in microgravity caused a number of artifacts early on, but once we started having good ventilation, forced air convection, those for the most part went away, not totally though. Another one is volatile organic compounds that are floating around the environment. And one classic example is Shannon Lucid when she was on Mir tending to the SVET greenhouse study. They were growing wheat, and they grew them, and they were looking pretty good, and they formed seed heads. And then I was at Kennedy when they brought them down. Everybody was excited, and they opened up the seed heads, and all of a sudden it went quiet because there were no seeds. Beautiful seed heads, but no seeds, and they did a series of studies on the ground, and they figured out that essentially it was an overabundance of ethylene on the Mir station. It's on the order of 1 parts per million, and ethylene is an endogenous plant growth regulator that can have all sorts of effects on plants. And they were actually able to pin down the high levels of ethylene to the periodic arrival, from the progress in the shuttle missions of fruit, which were producing and giving off ethylene. Next slide. So what are some of the key questions for the gravity environment? How does it affect the shape and alter the way plants grow and reproduce? Gravity directs root and stem growth. So in order for this to happen, you need gravity perception. You need to transfer the information from the site of perception to a site of reaction. And so NASA has sponsored studies, a lot of them on the ground, to identify the gravity perceiving cells. The threshold values, some of them have occurred in microgravity, and how the gravity vector is perceived. Next slide, please. Types of tropisms for plants. There's gravitropism, and for shoots, if you were to put this case, a plant horizontal, it'll curve up, okay, actually away from the gravity vector. There's also root gravitropism. It goes down towards the gravity vector. It makes sense. Shoots are looking for light. Roots are looking for water, which is generally down. Phototropism. Okay, you've seen that every day in your homes with your potted plants. You have to turn them. They keep bending towards the light. And hydrotropism. Roots will grow towards the water, actually search it out. There's currently a series of experiments that John Kiss is doing looking at phototropism in the microgravity of space. And the nice thing about it is it's often dwarfed by gravitropism on the Earth. Phototropism is a weaker force, so you fly it up into space, you remove that confounding, obfuscating factor, and you have a better way of looking at phototropism by itself. Next slide. So generally, there's a gravity stimulation, and I'll show you later ameloplasts, which are little dense organelles that settle down and initiate, not always, but often, the gravity response in plants, and it sets off a cascade of events involving calcium and calmodulin and another plant growth regulator auxin, which ends up with a selective cell growth. And it's actually what's happening is you're getting excess growth or enhanced growth on this outer part, so the shoots are growing up. And for the roots, you're getting enhanced growth on this side, so the roots are growing down. Next slide. So here are some ameloplasts in a normal root tip. You can see they're starch-filled organelles. They fall to the bottom of the cells and initiate the whole cascade of events. And people have done lots of studies with mutants that don't have them, or they have very less starch in them, so they don't have quite the response. Next slide. We saw Neil Pellis mention secondary metabolism. Plants have primary metabolism, what happens as a result of photosynthesis with the production of carbohydrates and sugars. Some of those compounds get shunted into what we call secondary metabolism that can help the plants in all different kinds of ways. They can make the plants taste bad or even be poisonous so that herbivores are dissuaded from munching on them. Well, there was a study with brassica grown on the ISS and fixed on the ISS. And by the way, you'll see little numbers. These refer to references that are provided at the back of the presentation. This work was done out of the Musgrave lab, but nominal growth and leaf chlorophyll, starch, soluble carbohydrates, so primary metabolism in this study was relatively equivalent between the spaceflight and the ground controls. But this secondary compound, glucosinolate, was about 75% or greater in the microgravity flown plants. Now, sometimes people look at microgravity grown seeds and they also see altered biochemical makeup compared to the ground controls. So, for instance, you can have, in some cases, and different experiments show different things. There's no one dogma for any of these results. But chlorophyll, starch, soluble carbohydrates increased, protein went down, storage reserve deposition went down. Another thing that you often see in terms of embryos of plants that are grown in space is that the range of developmental stages is much wider. Okay, it's not quite as uniform or compressed as what you see at 1G. So spaceflight clearly influences metabolite production in ways that affect plants, but in terms of kind of a practical way, it can affect the flavor and the nutritional quality of potential space produce. Next slide. So seeds, you can think of the effects of microgravity on seeds in the long term and in the short term. And one of the best studies for long-term exposure was on Mir with tomato seeds that were up there for 6 years. And they came back and they compared them to ground control seeds that didn't have microgravity exposure. And they found significant differences in growth and development relative to the ground controls. And also there were changes in the DNA. It's a bit more controversial for short-term exposure to seeds. There were a series of papers that came out of the Chinese space program that claimed genetic mutants from seeds that were flown for just short duration experiments also gave rise to strains with greatly enhanced productivity. But these results haven't been repeated or confirmed by American or European or Russian collaborators. Next slide. Movements, plants move, and there are some subtle movements like circumnutation, kind of a spiral movement, and early on there was a question as to whether that would happen in microgravity without a gravity vector to direct it, and the answer is yes, it still occurs. It's slightly different, so there are some interesting results, different patterns, differences in the transitions between dark and light in these movements, and some are heretofore unknown movements. Next slide. So what about pollen and seed development in space? And a lot of this work came out of Mary Musgrave's lab. But early on, you kind of have at the top here plants, pollen, and the female flower, the male part of the flower that didn't develop so well versus those that did. A lot of the earlier studies didn't have good forced air convection, So, you know, they were even in sealed containers, some of them. So you think of a plant without forced air convection, it immediately depletes the oxygen and or CO2 immediately adjacent to the plant tissues, so you end up with boundary layer problems. But when you do force air to flow past the stems or the roots or the leaves or whatever, that's gotten rid of. All right, so you can get rid of a lot of those artifacts by good forced air convection. Now, having said that, plants in microgravity still have differences, even with good forced air convection, with seeds and pollen that is often physiologically younger than the ground controls. And there's a speculation that even though you're forcing the air past the tissues, you're not really affecting what's happening within the seed pods themselves. So within the seed pods, you still have a lack of convection, so you can still have this depletion and boundary layer phenomenon that occurs and stunts your pollen and seed development. Next slide. There's an interesting result. It's actually some things that we found in 3 different experiments with 3 different plant systems. We've gotten enhanced root production in our microgravity grown plants, both in terms of the primary roots, the lateral root length, and the number of lateral roots that are developed. So why does this occur? There could be a physiological basis, or it could be one of these indirect spaceflight-associated artifacts. So for instance, we saw earlier from the SVET data that there's a kind of a shift upward in the moisture distribution within the root zone. Well, you could say that, well, maybe you have a more conducive environment for root growth in general as a result of that. Or there could be a real physiological microgravity effect, and there is a reference here, number 29, 30, where they looked at Arabidopsis that was germinated and fixed in space, and when they got it back and compared it to the ground controls, the root cortical cells were found to have proliferated at a higher rate, and they believe it may have been as a result of an accelerated cell cycle on the part of those cells. There's another result that we found at one time was a difference in the nutrient uptake of plants grown in space, where they took up twice as much potassium relative to the ground controls, while all the other nutrients were the same. Again, it could be a space flight, a real microgravity phenomenon, with some physiological basis, or it could be related to the enhanced root production, so there was a greater need for potassium to be taken up by the roots. Next slide. People have fixed plants in space for years, and the question is, do microgravity-grown plants exhibit alterations in ultrastructure? And the answer is sometimes yes. So they're giving you some references for mitochondria that appear to be different in microgravity versus their ground control, and even the vascular structure. People who have looked very closely and looked at the microfibrils of the cell wall have found differences. Next slide. So in terms of root tips, here we have some of these ameloplasts that are involved in the gravity perception, and this is the normal case, a transmission electron micrograph of corn cortical root cell on Earth, and you can see it's just jam-packed with starch, which is the normal situation. But counterpart plants grown in space have oil droplets, lipids, that have been displacing some of the starch. So this is a shift in metabolic production in space. Next slide. There's some classic work by Abraham Krikorian, who I was privileged to work with on several of these experiments, growing different kinds of plants, monocots, dicots, fully differentiated plants, and somatic embryos in space. And what he always found was chromosomal aberrations in the dividing cells, usually on the order of about 3% of the dividing cells, as found post-flight at the first metaphase, were found to have had chromosomal aberrations of one kind or another. This wasn't a showstopper. The plants dealt with it. Okay, they push those cells aside. It occurs in the meristematic zone where cells are rapidly dividing, so there was no real problem, although some of these came back and were grown out and they became aberrant in one fashion or another. Some of these were fixed on orbit, so here are these somatic embryos that were fixed on orbit to demonstrate that it wasn't a readaptation post-flight artifact. So these things happen, and they've been found in other systems as well, but again, the key point is it's not a showstopper. You can still grow plants through generations. Next slide. So also, as Dr. Pellis mentioned, there's been a lot of work recently with gene expression in plants grown in space, and almost always people find lots and lots of genes are being upregulated, others are being downregulated, so that's sort of the norm. There was a case where work by Furl and Paul found that some of these genes were related to heat shock, which he also mentioned. And so even though these were considered to be heat shock gene, the results couldn't really readily be explained by elevated temperatures. So remember, he mentioned that some of these so-called heat shock genes are really regulatory proteins, so they have other functions. Now there is one exception, the pesto experiment, which did preserve plants on orbit and looked at ground controls in terms of gene expression and found no significant difference. So clearly more work needs to be done in that area. Next slide. So also photosynthesis, respiration, transpiration. So these are kind of the day-to-day physiological processes that are key to successful plant growth. There was this experiment, the pesto experiment, where they grew plants in space. They formed pretty much a full canopy, and they measured photosynthesis by sealing off the canopy and having the plants draw down carbon dioxide as a result of photosynthesizing. So you can get a photosynthetic rate that way. And when they compared that to the ground control plants, there was no significant difference. But having said that, when they brought the plants back and they did more intensive experiments on a single leaf basis and kind of looked at what we call the Z-scheme, the light reactions of photosynthesis, there were differences, and this confirms some earlier studies that we had done back in the early 90s. So the bottom line is there are some subtle photosynthetic functions that are affected by the spaceflight environment, but again, they're not showstoppers, and the plants can keep on going despite what's happening. Next slide. So just in conclusion, we've learned quite a bit about growing plants in space. There's a lot more to learn. The shuttle is kind of flying off into the sunset, but we still have the International Space Station to tease apart a lot of these mysteries, and I think we should continue, and with the eventual goal, well, we'll learn something about plants on Earth, but we can also move towards bioregenerative life support system for when we have planetary surface, long duration planetary surface habitats. Next slide. Thank you. Yes, way back. Yes, among others. So the question is, I pointed out about the heat stress genes being upregulated. Are there other stress-related genes that were also found to be upregulated? And essentially this isn't my work, but the answer is yes. I can refer you to some investigators who have and are currently in the process of publishing those results. One more question.
That's the first time I heard the presentation on plant growth in detail. Has anybody looked at the GM foods as such, genetically modified foods, and how they respond to the microgravity environment?
The question was whether any genetically modified plants have been grown in space and how they respond to the microgravity environment. And to my knowledge, the answer is no. There have been strains that have been developed, the super dwarf wheat, for instance, perigee and some others, but they have been developed by traditional plant breeding techniques, not genetically modified techniques. Thank you.